All right, welcome to my video on the topic of muscle plasticity in response to use and disuse. This is based off of chapter 21 in Hill, Kavanaugh, and Anderson's Animal Physiology text. The key points we're going to cover in this video are the idea that muscle is a dynamic tissue, so muscle size is a product of the relative rates of anabolic and catabolic activities taking place. We're going to look at how muscle power output determines its contractile performance and how power output changes in response to use and disuse. We're going to review how a training stimulus type will determine how muscle responds and the different signaling pathways that are involved in responses to different types of training stimuli. We'll investigate atrophy and the idea that age and disuse are commonly associated with atrophy processes. And over the course of studying muscle plasticity, we're going to be introduced to two different signals that are involved, signaling factor that's really important, and pathway that's involved in muscle tissue dynamics. So we're going to visit these points as components of a four-part story. First, we're going to investigate muscle hypertrophy in response to endurance training. Then we'll examine hypertrophy in response to strength or resistance training. I'm going to share an amazing story with you of hypertrophy and atrophy, and then I'll talk more specifically about atrophy and mechanisms that determine muscle mass. So again, muscle is a dynamic tissue. So another way of phrasing this is just to note that it's very phenotypically plastic. And because it's a dynamic tissue, basically muscle mass at any given point in time is going to reflect kind of a balance between anabolic processes, or processes that build muscle tissue, and catabolic processes, or processes that break down tissue. So when muscles undergo net anabolic processes, they'll experience hypertrophy, where if they experience net catabolic processes, they will then undergo atrophy. And so to understand how muscle changes, we have to learn a little bit more first about our different muscle fiber types, which I introduced in a previous lecture. So just as a quick reminder, there are three major skeletal muscle fiber types, and they differ in terms of their function, density of mitochondria, the capillary supply, their twitch speed, fatigue, also their myosin and calcium ATPase isoforms as well, right? And I should note here that previously I've talked about how metabolic enzymes typically within an individual, part of the acclimation response to changing conditions involves changes in the amount of metabolic enzymes if we're looking within a species, within an individual, right? And we only see changes to isoforms of metabolic enzymes when we're looking across species. With muscle proteins, in contrast, right, there are different distinct isoforms of these proteins, right? So there's the myosin that can relatively quickly hydrolyze ATP. There are faster calcium ATPase, calcium uptake proteins, and then there are slower forms of these proteins, right? So the function of any particular muscle fiber is going to be a product of which particular isoforms of myosin and calcium ATPase are present. Right. So again, I've highlighted, our three major fiber types include slow oxidative, right, which are well vascularized and have larger internal stores of myoglobin, right? So, but as the name implies, are relatively slow to produce force. Then, in contrast, fast glycolytic fibers, right, depend predominantly on anaerobic ATP production, right? So they're very fast. They have a wider cross-sectional area, different myosin isoforms, and then there's an intermediate fiber type known as fast oxidative glycolytic, which to some extent we can think of as being intermediate in properties between slow oxidative and fast glycolytic. Throughout this lecture, I'm going to, rather than using the general terms for muscle fiber types, slow oxidative, fast oxidative glycolytic, or fast glycolytic, and you can see they're abbreviated here, I'm going to use the human terms for them, where in human physiology research, slow oxidative are referred to as type 1, fast oxidative glycolytic are referred to as type 2A, and then fast glycolytic are referred to as type 2X. Right, and this is just a summary table to remind us of differences in myosin properties and differences in the calcium ATPase properties, right, which correspond with what I've already explained to you. So taking a little bit of a step back, just to reiterate, that the major job of muscles and skeletal muscles is to perform work. And from a physics standpoint, right, you can quantify work as the product of the force that's produced and displacement. Okay, this then leads to a question of, okay, well, how can a muscle generate more force to do more work? And the major way that muscles can accomplish this is through the presence of a larger cross-sectional area, right? So if a muscle has a smaller cross-sectional area, right, compared to a muscle that has a larger cross-sectional area, a smaller muscle, by definition, will wind up having fewer myosin and actin contact points. So force production will be lower, okay? Where if a muscle has larger cross-sectional area, there's physically more space to fit in thick and thin filaments, so it's possible to generate more force. And that's the, basically law of physics at work right there. Let's look at something else as well. So if you have two muscles that have the same cross-sectional area, but one muscle is twice as long as the other, if I were to ask you which of these muscles would do more work and why, I'd suggest taking a second to just pause this video and think over which of these you'd expect to do more work and why. And I'll talk about the answer momentarily. Okay, so the answer is the muscle on the right that's twice as long would do more work, right? And in this case, both muscles would be able to produce equivalent amounts of force, but if you have a muscle that's twice as long, right, it's going to undergo twice as much displacement over the course of a contraction cycle. All right, along with thinking about force, we should think about the quantity of power. Right, so I'm just going to re-abbreviate over here. Work is force times displacement. Power, from a physics standpoint, is measured as the amount of work conducted over a period of time. Okay, so it's the rate at which work is being conducted. Okay, along with that, we should think about velocity, right, which is a measure of the amount of displacement over time. Okay. So if you look at these, work, power, and velocity, I hope you can appreciate that there's an interrelationship here between them, right? So you see displacement and time showing up across all three of these. So if you do the fancy math and do some substitutions, you can then quantify muscle power as a product of force times shortening velocity. Okay, so that's a typical unit for characterizing muscle power. So generally speaking, if you're trying to characterize muscle's contractile performance, it's then measured as that muscle's power output, right? Measured as a product of force times velocity. So I have a kind of a complex graph here that I want to take a second to walk through so that we can understand how to interpret muscle power output. So the x-axis on this graph is force, right, in units of newtons. Okay, so on the left-hand side, low force, on the right-hand side, high force. And then the y-axis here is the muscle velocity, right, meters per second. So low velocity down towards the origin, high velocity up towards the top. And what I want you to focus on right now is what happens to velocity relative to the amount of force that's being generated, okay? And so this blue line here is that relationship, right? So as the amount of force that is needed increases, right, velocity will decrease. And so if we look down here at this far right-hand point, I've highlighted this is where you'd observe an isometric contraction. Velocity is 0, right, because muscle's not shortening. And this is because the load that the muscle's working against is equal to the maximal force that that muscle could generate. So this is where you'd see purely an isometric contraction. At the other extreme, if the force that is needed is minimal, so there's no load for that muscle to be acting against, that's where you'd wind up seeing the maximal possible velocity for that muscle. So this actually asymptotes at a force of 0. Okay. Again, because muscle power is a product of force and shortening velocity, if we map out muscle power as the product of those two elements, you wind up getting this red curve that's illustrated here. Right? And the main point that this highlights is that a muscle's peak power is usually going to take place at about 20 to 40% of that muscle's maximal velocity, right? So right here, 20 to 40% of that. And it will be at about 30 to 40% of the maximal load, the load that keeps it from shortening. So that's, sort of setting our standard for muscle power, muscle peak power. So then, the question comes up, well, how might muscle power change? How could it increase? I've highlighted one way that that can happen, right, which is through an increase in a muscle's cross-sectional area, right, because then there's more fibers that can engage, have more myosin heads engaging with actin. But along with that, power can also change as a result of a change to the muscle fiber type, okay? For changes to happen, however, the type of stimulus that a muscle receives will also impact how that muscle responds. And so we're going to divide our types of stimuli into our two major categories of exercise. So endurance exercise, right, which takes place at submaximal rate relative to VO2 max, versus a resistance or strength type stimuli, right, which are what take place when an animal is engaging in supramaximal exercise, right? So that's exercise that winds up being self-limiting because it's above VO2 max. So muscle tissues are not receiving oxygen at the rate that is necessary to generate ATP. To appreciate how endurance training can alter muscle fiber types, a lot of this work comes from studies that involve taking muscle tissue biopsies. So just kind of illustrating how a lot of this work is conducted. So here's a muscle tissue punch. So typically participants who engage in these studies will engage in some sort of exercise treatment, and then over the course of that exercise treatment, so a muscle tissue biopsy will be taken so that physiologists can examine changes to muscle fiber types. And they will typically be given a local anesthetic before the tissue biopsy is taken. And most commonly, tissue samples will wind up being taken from the quadriceps because that's one of the largest muscles in the body. Okay, so here's a chart from our textbook that highlights major changes to muscle as a result of endurance training. And so this is broken down according to the major 3 fiber types, and also highlight some differences between men and women. So on the left-hand side, we've got characteristics before individuals experience endurance training, and then on the right-hand side, we've got characteristics after endurance training. And this is highlighting both muscle fiber types and then also the density of capillaries that are present around muscle. So a couple of things I want you to notice from this table. On the left-hand side, first of all, notice that the predominant muscle fiber type that's present, although it varies between men and women, is primarily type 1 fibers, right? So again, type 1 are the slow oxidative fibers. Type 2A are the fast oxidative glycolytic fibers, and then type 2X are the fast glycolytic fibers. Okay, so notice in women that initially type 1 fibers predominate in muscle. Okay, and this is actually a little bit high. I'll highlight that in just a moment. And the next most prevalent fiber type are type 2A fibers, and there's a little bit of type 2X fibers present within muscle. In men, there's a slightly more even distribution between type 1 and type 2A, but still type 1 are the most common. Type 2A intermediate, and then type 2X are the least common. And we won't dwell too heavily on capillary densities, but you will notice, right, they do vary, as I indicated, right? So type 1 and type 2A tend to have higher capillary densities than our type 2X fibers do. Okay, so now after training, the biggest change that takes place is actually a shift away from type 2X fibers to type 2A fibers, okay? And right, notice that that takes place both across men and across women, okay? Okay, so endurance training is really leading to a shift from fast glycolytic to fast oxidative glycolytic. So there's not really a major change in slow oxidative fibers in response to endurance training. Okay, so what this is highlighting is that type 2X fibers appear to be more plastic than type 1 fibers do. Okay, I'm going to reserve comments on type 2A for the time being. Okay, you also notice there's an increase in capillary density around all of the fiber types after training. And along with this, across muscle fiber types, you'll also observe an increase in mitochondrial density. So generally speaking, although these are categorized based on the major method that's used to generate ATP, they can all engage in some degree of aerobic metabolism. And so generally speaking, characteristics that facilitate increased oxygen delivery are upregulated in response to endurance training. And this should kind of come as no surprise based on what we talked about earlier in terms of changes to oxygen delivery and VO2 max in response to endurance training, right? So this would facilitate improved oxygen delivery and facilitate an increase to VO2 max in response to endurance training. Okay, yeah, so just one quick note on the type 1 fibers in women. This value is actually unusually high for women across a larger range of studies, so some participants wound up dropping out of this experiment, and they had a type 1 fiber density that was closer to about 47%. So again, as is typical, I'm not going to expect you to memorize the specific numbers within this table, but I do want you aware of the fact that the major shift that's taking place in response to endurance training is a shift away from type 2X and towards type 2A fibers. Okay. Along with those changes, there will also be a change in the amount of lipid droplets that are stored within the muscle. And this suggests that in response to endurance training, muscles will shift towards a greater reliance on fatty acid oxidation for energy supply compared to individuals that have not undergone endurance training. Right? And that's just highlighted again in a graph from our textbook. Here, so again, showing the increase in mitochondrial density and then an increase in lipid droplet content, right? And to oxidize lipids, cells do need to have a consistent oxygen supply in order to use lipids as a major energy source. Okay, so how is it that the capillary density is increased? The major element contributing to that is production of a growth factor known as vascular endothelial growth factor. So conveniently, it's named after what it accomplishes. Okay, so the data in this figure come from a study that compared trained versus untrained muscles and then measured expression of mRNA for VEGF. And this showed that following exercise, right, so the red bars here are at rest, the blue bars are immediately after exercise, there will be an increase in VEGF amounts. But the thing to notice here is that VEGF amounts actually don't increase as much once muscles are more highly trained, right? So upon initial exercise, this just suggests that initiating endurance training, you'll see a much larger change to VEGF and a much larger change to vascularization of muscle tissue. But once training has commenced, although you still see an upregulation in VEGF, it's not quite as extensive. So there's kind of an upper limit to the extent to which angiogenesis can happen, right? Because again, we're looking at a tissue where space is really at a premium. And I just want to point out that this data comes from a really clever experiment where, to control for individual differences in some of these responses, they worked with individuals, so they had people exercise one of their legs but not the other, right, to highlight this. Okay, so I also want to point out some of the changes in gene expression, right, that facilitate the changes that are seen during endurance exercise. This work comes from studies of mice that have had a key gene knocked out, which helps us to determine that the critical factor involved in leading to this change away from type 2X to type 2A. So in these studies, wild-type mice were compared to MKO mice, which are knockout mice that don't have a gene called PGC1-alpha-1. And they only fail to express PGC1-alpha-1 within skeletal muscles, okay? So if we look at the graphs here down below, this highlights average activity levels for both mouse types, right? So I hope you can see from this graph that there's no major difference in the extent to which these mice are active. But if we look over here on the right-hand side at capillary density and changes to capillary density in response to exercise, what I hope you can see here, so sedentary mice have very similar capillary densities. Wild-type mice, unsurprisingly, have high capillary densities, but in the PGC1-alpha-1 knockout mice, we see a similar capillary density across these two treatment groups. So this suggests that PGC1-alpha-1 is a critical signaling factor leading to changes that are associated with endurance exercise. So I'm going to say key signaling factor stimulating changes in response to endurance exercise. Okay, so although we see these changes in muscle tissue during endurance training, you might know this already, we don't really see any change in muscle mass. So there's a change in fiber type, but not a change to muscle mass. So what is it that actually leads to an actual change in muscle mass? That occurs in response to resistance or strength training, okay? And I want to highlight that the hypertrophy that is observed in response to resistance or strength training is not the same thing as hyperplasia. So hypertrophy refers to an increase in muscle size or muscle mass, but not the number of muscle cells. Right, so which is what hyperplasia refers to. So it's important to keep in mind that typically the number of muscle fiber cells that are present within a muscle remains constant, generally speaking, over the lifespan of an animal. So only the size of muscle fibers changes. So interestingly, so resistance training it leads to this outcome of hypertrophy, so larger muscle fiber size, larger muscle fiber cross-sectional area, right? So an increase in potential force production. And it also leads to a change in fiber type. And perhaps paradoxically, what happens during resistance training is also a shift from type 2X towards type 2A fibers. However, for resistance training, the signaling pathway that is involved is different than the signaling pathway that stimulates this shift during endurance training. So for resistance training, the major signaling factor that's involved is a slightly different PGC1-alpha-4, okay? So here's a summary figure from our textbook that's just highlighting the signaling pathways and the major changes that occur in response to endurance versus resistance exercise. So just to reiterate, during endurance exercise, there will be an increase in PGC1-alpha-1. That's actually going to favor production of myosin heavy chain types, isoforms type 1 and 2A, right, which facilitate sustained force production, increase in mitochondrial densities, an increase in fatty acid oxidation, right, linked with the increased lipid droplet storage, and then an increased production of VEGF, so stimulating an increase in capillary density. And then by comparison, during resistance exercise, there will be an increase in PGC1-alpha-4 signaling. That will lead to a decrease in a factor known as myostatin, which I'll talk about a little bit more in a little while, and also an increase in insulin growth factor 1, which will stimulate muscle hypertrophy and an increase in muscle strength. All things considered, right, if you're looking at this sort of thinking about, well, what makes the most sense for my muscles? At the end of the day, usually it's best to engage in a combination of both types of training, right? There are a lot of benefits to endurance training, but there are also a lot of benefits to resistance training. And collectively, both forms of exercise can improve overall performance. Okay. Now I want to take a minute to walk you through a really incredible story of hypertrophy and atrophy. And for this story, I want you to appreciate that cardiac muscle can also increase in size through hypertrophy. Okay, so for this story, we're going to consider an animal, the Burmese python. Okay, and some of the images that I'm going to show you for this portion come from a study that was published in 2008. So I've given you the reference here at the bottom. So Burmese pythons are typically fairly mellow animals. But they have an interesting lifestyle where occasionally they will eat things that are up to 50% of their body mass. And I should also note that periodically you may see stories in the news about Burmese pythons because they were very popular as a type of pet, but as often happens, when people keep animals in captivity, a lot of people that have kept Burmese pythons discovered that they can get really, really large. And as they get large, they become unwieldy to keep as pets. So they wound up being released in all kinds of places. And they're now actually a huge problem in the Florida Everglades because they're willing to eat all kinds of things, and they're a substantial predator type. And on occasion, you'll see stories about Burmese pythons trying to do things like eat alligators, just to give you a sense of scale. So in any case, most of the time, they just wind up hanging out, but periodically, they will eat an enormous meal. And as I talked about when we were talking about energy metabolism, right, the process of digesting a large meal is a very energetically intensive process, right? An animal has to produce enzymes that will break down the nutrients that are in the food that it's consuming, and you can measure that. If you measure an animal's metabolic rate using indirect calorimetry through respirometry, and you'll see something known as the heat increment of feeding, a big spike in metabolism while that animal is digesting. Well, to make that happen, a python also has to increase its heart volume and its circulatory function, right, to deliver enough oxygen to tissues to produce those enzymes to break down that meal. So while a Burmese python is digesting, its heart ventricle will increase by 40%. So that's a massive amount of hypertrophy, right? So this is an image from that paper, first just showing us tissues in a fasted python relative to tissues in a fed python, right? And the structure over here on the right-hand side is the python's heart, right? So I hope you can appreciate this is major, major hypertrophy. And I also want to note that, the physiologists that were studying this hypertrophy in pythons, they're just interested in understanding how on earth is this accomplished, because this is pretty incredible. Okay, so over the course of studying this, they observed that the hypertrophy of the heart is linked to an increased production of messenger RNA for cardiac myosin heavy chain, right? So a particular myosin isoform found in heart muscle, okay? And so here's a couple of different measurements collected in these digesting pythons, right? So the ventricular mass is on the left-hand side here, so hopefully you can see, right, here's the ventricular mass when fasted, here's the ventricular mass post-digestion. So during digestion, the mass of the ventricle in pythons has increased, right? Okay, here again is what happens with the myosin heavy chain mRNA. So again, it's much, much higher, so that indicates increased production of heavy chain myosin, the cardiac isoform, okay? But interestingly, if you go and measure the amount of DNA that's present per unit of muscle mass, it actually is lower. Right, and that indicates that there's no production of new cells associated with this increase in cardiac muscle mass. So again, that just reemphasizes the point that when we see changes in muscle characteristics, those are primarily just changes within muscle cells, not changes in muscle cell proliferation. Okay. So, then they're curious about, okay, well, how is this hypertrophy actually accomplished? The scientists studying this discovered that the signal that leads to cardiac hypertrophy is actually a blend of 3 fatty acids. So here on the left-hand side, I've got pythons after a range of treatments, and the y-axis here is % change in heart mass. So on the left-hand side, this is 3 days post-feeding, okay? So again, showing hypertrophy, okay? The next bar here is the cardiac muscle size in pythons that were injected with plasma from a fasted python. Okay, so notice if a python is fasted and receives fasted plasma, you don't see this major hypertrophy response. However, when pythons were injected with plasma from a python that had recently acquired a big meal, we see this type of hypertrophy response. So there's some signal present in blood plasma that's leading to this level of hypertrophy. If you inject pythons with just a vehicle, so this is a control treatment, you don't see hypertrophy. But if you inject pythons with 3 fatty acids that were derived from the plasma of fed pythons, you see the same type of response and the same degree of hypertrophy. So the figures here just again reiterate some of those changes. Okay. The coolest part of this is after discovering that this blend of fatty acids leads to cardiac hypertrophy in pythons, the scientists also figured out that this same signal of 3 fatty acids can generate cardiac hypertrophy in mice and rats as well. So not only did this reveal insights into muscle function in pythons, but also is something that is more broadly applicable to other types of animals. And actually, this can also potentially work in humans as well. So I've included a link here at the bottom of this slide to a quick article that talks about how this discovery in pythons could be really useful for treating circumstances of human cardiac disease as well. I do want to highlight here that in some cases, we can think of hypertrophy as being a good thing, right? So as an animal develops, it needs to be able to produce more force, if it gets larger. During pregnancy, cardiac hypertrophy can be really important for delivering blood and nutrients to developing embryo. And under other circumstances, it can also be important. So for example, when an animal finishes hibernating, it goes from a condition of dormancy where it does not need to be able to move around to now needing to be able to move around its environment again. But it's important to keep in mind that there can be circumstances where hypertrophy is not beneficial. So we need to distinguish useful hypertrophy from pathological hypertrophy. So for example, hypertrophy in the absence of other changes, will just lead to high blood pressure and heart attacks. Along with that, in some cases, hypertrophy can be associated with cell death and a shift away from lipid to glucose oxidation, which is accompanied by an increased mortality risk. However, discoveries from animals such as the pythons are fantastic for helping us to develop better therapeutics for dealing with these types of muscle conditions. Right, so that brings us through hypertrophy and changes in response to exercise. So now we're going to shift gears and we're going to talk about atrophy. And just as a reminder, right, we observe muscle hypertrophy in cases where anabolic processes are occurring faster than catabolic processes, right? So muscles are dynamic tissue where there's turnover, and so the relative rates of anabolic processes and catabolic processes determine whether a muscle is experiencing a condition of hypertrophy or whether that muscle is going to be experiencing a condition of atrophy. So during processes of atrophy, perhaps unsurprisingly, there will be an observed change in fiber size, right? So that indicates that more of the sarcomeres are being broken down. And along with that, there will be a change in fiber type. And typically speaking, the major change that's observed during atrophy is a decrease in type 1 fibers, okay? And the primary increase that's observed is an increase in type 2X fibers, okay? So on our little chart over here, here are the major consequences of muscle atrophy, okay? The major factors that tend to contribute to atrophy are disuse and age. Okay, so here are some muscle tissue biopsies from astronauts, right? So they spent some time in space where they're not experiencing gravitational forces, right, which are very important for physiological work. So before launch, what I want you to notice, right, this is just illustrating in a slide one sarcomere within muscle. And notice, right, that this is standardized to the length of the sarcomere. So you can see sarcomere length on the left-hand side, also sarcomere length on the right-hand side. But along with length, notice differences in the width of these sarcomeres. So it's much wider before launch. 17 days after spending 17 days in microgravity, sarcomeres are much, much narrower, right? So indicative of atrophy. This slide also shows us what some of these muscle lipid droplets can look like. And I will just note that astronauts that have spent even longer in space also show a reduction in this type of lipid accumulation within muscle, right? So that's another signature of atrophy. So that's a pretty vivid example of changes in response to disuse. But as I also mentioned, muscle atrophy will also occur over the course of aging. So here's a graph just showing age of men and women on the x-axis in years. And then on the y-axis, this is highlighting muscle performance across two contexts. So one is just relative to overall running speed, and one is running speed over the course of 3 different event lengths, okay? So there's a couple of major takeaways from here, right? So first, right, we see a peak in muscle performance. So peak running speed can be achieved somewhere around the early 20s. And then as humans get older, right, we're seeing a decline of running speed under these conditions. On the right-hand side here, just looking at responses in men, I want to highlight that we see a peak in running speed for shorter distances at a younger age, and interestingly, a peak for endurance events occurs at a slightly older age. I also want to note that this highlights kind of only 2 aspects of muscle performance, and I think this is actually a little bit short-sighted because the performance peaks can very much differ based on sex, based on activity type, but also based on the method for how performance is being evaluated. Right, so here this is just purely focused on speed, but along with that, we could also characterize muscle performance in terms of recovery of power, how fatigable the muscle is, and overall endurance of muscle. And if we look across a wider spectrum of muscle characteristics, some of this simplistic picture starts to fall apart. And I bring that up primarily because to some extent this picture can be a little bit discouraging because it suggests that as you get older, your athletic career is going to come to an end. But in actuality, because this is only focused on one particular aspect of muscle function, it's a kind of a short-sighted snapshot of athletic performance potential across sports, across a person's lifespan. So although, for example, we might see a peak in some sports that really favor speed, right? So sprint speed in particular. So you might see those peaks at a much younger age. In other sports, we do know of examples of athletes that are still, striving for and achieving peak performance, well into their 40s. So I think there is some reason to still hope and think that muscle performance can persist over a greater portion of a person's lifespan. Along with this, I want to highlight that not all animals experience atrophy in the same fashion, right? So generally speaking, we see muscle atrophy in response to disuse and increasing age. However, interestingly, physiologists studying muscle characteristics in hibernating bears and also in burrowing frogs that are estivating, right? So they're in a dormant period to avoid very dry conditions. Right? When muscle characteristics are measured in these animals, physiologists see very little atrophy in key muscles for movement. Right? So this suggests that that disuse does not always lead to the process of atrophy. We only see atrophy in specific circumstances. And we might be able to learn something from signaling factors that are found in animals that don't undergo atrophy compared to animals that do. Okay, so last but not least, I want to talk about one more set of factors that are involved in regulating muscle mass. The major factor I want to highlight is a signaling factor known as myostatin. So myostatin is a signaling factor involved in regulating overall muscle mass. And we can see a really vivid example of the impacts of myostatin in a dog breed that has a mutation for myostatin production. So the whippet that's pictured here on the left-hand side is a whippet that's referred to as being double-muscled. And it has this characteristic because myostatin production is impaired, right? So that suggests myostatin production is important in reducing overall muscle mass, right, compared to whippet on the right here, right, which has much lower muscle mass and has functional myostatin present. And this might seem like a really good thing, but again, I want to note that there are reasons why hypertrophy might not always be a good thing, right? So although the double-muscled whippet may be capable of much greater power and production. At the same time, there are a lot of consequences for circulatory processes and overall animal health for carrying around this extra muscle mass. So in addition to the PGC1-alpha-1 and PGC1-alpha-4 signaling factors, the overall pathway that regulates the balance between synthesis and degradation within muscles is a pathway known as the P13K-AKT1 pathway. So to kind of give us a sense of how this works, as a muscle produces force, a muscle will then secrete IGF-1, so insulin growth factor 1, right? And IGF-1, in this case, will act as an autocrine signal on the muscle, right? So it produces force, squeezes out IGF-1. The IGF-1 is then detected by insulin receptors that are present on the muscle cell surface. IGF-1 will then activate P13K and AKT1. And when it does so, this increases protein synthesis and it blocks protein degradation. So when this is happening, so when a muscle is working, producing IGF-1, leading to the signaling cascade, protein synthesis rates will go up, and the transcription of genes that are associated with atrophy will decrease. Muscles will increase amino acid uptake and we'll see hypertrophy. Okay. In contrast, when muscles are not engaged in force production, so during periods of inactivity, the transcription of atrophy genes will be higher, right, because AKT1 is not active. We'll see a drop in protein synthesis rates, and the overall outcome will be protein degradation. Okay, and there you have it. So hopefully from this lecture you've come to appreciate how muscle is really a very dynamic tissue. Muscle mass is a result of a balance between anabolic and catabolic processes. Muscle power output will determine its contractile performance and can change and will change in response to use and disuse. The type of training stimulus will determine how muscle responds and recruits different signaling pathways to facilitate those responses. Atrophy is typically associated with age and disuse, but again, it's not always associated with that. Hopefully this has highlighted the involvement of two signals, so PGC 1-alpha-1 and 1-alpha-4, myostatin as a regulator of overall muscle mass, and the role of the P13K AKT1 pathway in regulating this balance between synthesis and degradation.